Welcome to What Did I Do in Lab Yesterday, where I tell you guys what I did in my biochemistry lab yesterday. So, what we did yesterday was gel chromatography. If you don't know what that is, I will tell you, I guess. So, gel chromatography is one of the methods used in order to separate protein based on their sizes. So what you do, you get this gel medium, which is basically a bunch of really tiny Orbeez packed into a glass tube. Then you put a sample of a bunch of protein mixed together onto that tube, and you put some buffer on top and wait for it to drain. This is the gel chromatography tube. All of the stuff in there, that opaque stuff, is the gel medium, and the colored stuff is the protein that is in the process of separating. So how it works is you put the sample on the top, and the proteins that are smaller will get stuck in the pores of the gel beads and stay in it longer, and the proteins that are larger will not get stuck in the little pores, and they'll drain through the column a lot faster and come out first. So that means the red stuff on the very top of the gel is the smallest proteins, with the yellow stuff being slightly bigger, and you can't really see it, but the stuff on closer to the bottom is slightly bluish, and those are the biggest proteins. As it drains through, it then gets dripped into these little cuvettes, and we collect about a milliliter of it, and we collect them in the cuvettes and put them in a spectrometer. Is that it? Okay. It measures the absorbance of different wavelengths of light based on the basically color and what color the proteins absorb. And then you can basically tell what protein is by the wavelength of light that says is absorbed by the spectrometer. And what's really fun about gel chromatography is that it takes fucking forever. I was in lab for almost 4 hours yesterday. Half the lab was waiting for the gel beads to settle and get packed in correctly so the protein would drain through correctly. And then the other half of the lab was watching the protein go from the top of the column all the way down to the bottom of the column, excruciatingly slow. And then measuring the absorbance in the different cuvettes. It took so long. We were sitting there for 4 hours watching the color go slowly down the column and adding more buffer and collecting it, but it was so fun.